Hello everyone. In today's presentation, I'm going to discuss about the first project of my PhD, which is titled Age-Related Increase in the Locus Coeruleus Activity and its Connectivity with the Prefrontal Cortex during Ambiguity Processing, where we focused on understanding how the locus coeruleus supports emotional processing as we age. To start with the introduction, we know that the emotions are fundamental for social interactions, and especially they are at the heart of how we connect with others. They not only help us to understand and relate with other person, but also build trust in everyday interaction as well. Even more critical for social interaction is interpreting those emotions accurately, as it helps us to respond with empathy and build again genuine connections as well. But not all emotions are clear in our day-to-day life. Many can be subtle or even ambiguous as well, which can make it extremely difficult. But at the same time, even more important to interpret them carefully to improve our connection with the other person. And especially as we age, interpreting these ambiguous emotions can require additional cognitive effort and but it still remains unclear what are the adaptive neurobiological mechanisms that support this process. And one of the prime candidates in this process could be the locus coeruleus. It's an extremely tiny brainstem structure, probably twice the size of the rice, and it is a home of noradrenaline as well. Some of the important functions of this region is regulating attention, arousal and vigilance, encoding salient stimuli, and also in responding to stress and processing uncertainty as well. And how LC function is that the LC activity follows an inverted U-shaped curve, as you can see here in the figure, where too little activity, as you can see here, can lead to inattention, while too much can also cause distraction. So for optimal focused attention, the moderate tonic phasic activity is essential, like everything in the world, where balanced activity can help you in everything. And especially, the locus coeruleus also projects to the cortex, and it has especially dense projection to the prefrontal cortex. And most critically, top-down regulatory mechanism of PFC coming back and projecting to the brainstem regions are also critical for resolving this conflicting information as well. And more recently, it has shown to be linked with inhibitory control as well. And how prefrontal cortex function best is at moderate levels of noradrenaline, and it has been seen in the rodent literature as well, where, as you can see in the green, the optimal PFC functioning has high level of noradrenaline level, and it helps in the top-down control as well. And not only that, recent studies have also found some links with with this optimal PFC functioning with emotional regulation, anxiety, and regulating stress as well in again rodent literature mostly. Regarding the human literature and especially human aging literature, it has been seen that the preserved LC structure, locus coeruleus structure integrity, is associated with better episodic memory in late life as well. And most importantly, elevated locus coeruleus activity. So not only the structure part of the locus coeruleus, but the functional activity that we see in the fMRI in the bold blood oxygenated level dependent response is generally higher during novel phase encoding stimuli. And this has been linked with greater resilience against cognitive decline in late life, as you can see in the Prokopis et al. Study. And more recently, Ludwig et al. Also found similar results where during the emotionally salient stimuli task, they found higher locus coeruleus activity in the older subjects as well. And this indicates the context-specific adaptive engagement of the locus coeruleus in late life and its potential link with cognitive decline as well. But its link with the mental well-being in humans, and especially in the late life, remains unclear. So, To answer that, we have two main questions and predictions in our study. The first question is, what is the role of this locus coeruleus activity, especially during the encoding ambiguous facial expression in late life? And we predict based on the prior literature of Prokopis and the et al. Study and the Ludwig et al. Study that there might be age-related increase in the locus coeruleus activity as well as processing ambiguity requires more cognitive effort. There might be higher adaptive locus coeruleus activity in older subjects compared to the young one. Next, we also try to answer whether the locus coeruleus connectivity with the prefrontal cortex during ambiguous facial processing and whether it links with some sort of emotional well-being in late life. And we predict that there might be again adaptive role of prefrontal locus coeruleus PFC connectivity as well during ambiguity processing in late life. And to achieve this, we analyzed data of 75 younger subjects and 69 older subjects, age ranging 65 and 85 years, in our Trondheim Aging Brain Study, which is based on the 70 MRI, where participants completed a task where they see a face, where the faces are morphed across 7 distinct levels, such as the face could be either 100% fearful or 100% happy, so clear and distinct emotions in it. And to manipulate the ambiguity condition, there are other 5 conditions. So the face could either be morphed in 70% fearful condition or 30% happy, 60% fearful or 40% happy, and so on in the opposite side. So 60% happy or 40% fearful. And most critical condition was in our study was the 50-50 absolute ambiguity condition, where the face was morphed 50% to the happy and 50% to the fearful condition. For more information on how we achieve this morphing, feel free to scan the QR code for the preprint. But again, to continue, so after the participants see the face or encode the face, they make a decision whether the face was happy or fearful. And after that, they also respond how confident they feel about their decision that they made earlier. So they either select they're not sure, sure, or very sure about the decision they have made. And apart from the MRI data, we also have the behavioral data where participants completed cognitive behavior tasks such as the emotional recognition, memory, and certain executive functioning tasks, such as truth tasks. And they also completed self-reported mental well-being questionnaires as well. So for example, standardized measures or questionnaires of depression, anxiety, stress, resilience, emotional regulation, and so on. Again, for more information, feel free to check the QR code. But since we had many of the measures, we decided to run a principal component analysis where we have, after we ran the PCA on it, we got the composite scores of each of the measures, the cognitive and mental well-being measures separately. And based on that, we derived 2 major indexes that we used in the study, which is the cognitive reserve index and the emotional resilience index. For the locus coeruleus, we used the template-based delineation. So we had the high-resolution magnetization transfer image, as you can see, extremely tiny voxel size of 0.4 by 0.4 by 0.5 millimeter. And we had the individual empty images as well. And first, we did some cleaning where we normalized the individual images by referencing the pontine tegmentum region, as you can see in the light blue color, which is anatomically close region to the locus coeruleus, and it helps us to reduce the intersubject variability that we see. After the intensity normalization, as you can see in C, we did the study-specific empty template. We created it based on the ends function. And after that, we delineate the left and the right locus coeruleus on the template. But still, we have to use this locus coeruleus template for the functional analysis, and most of the functional analysis is on the MNI space. So to achieve this, we first linearly registered the empty template to the subject-specific T1 template. So subject-specific T1 template is uh, based on the T1 weighted images as well. And since MTTFL and automatization transfer images or the template and the T1 images were acquired in the same session and from the same individuals, a rigid registration was sufficient to ensure the accurate anatomical alignment while also minimizing the distortion. Next, the whole brain study specific template, T1 template, was non-linearly registered to the MNI space using the ENS registration function again. And why non-linear registration? Because it helps us to account for the individual anatomical variability and ensure proper standardization to the MNI standard space as well. As again, most of the functional images, as all of the functional images are on the MNI space generally. And finally, using the step 2 and step 3 transformation, and finally using the step 2 and step 3, we transform the locus coeruleus mask to the MNI space in one single step using the ENS transformation function as well. Behaviorally, we found the effect of condition such that during the absolute ambiguity condition, where the absolute ambiguity where the faces are morphed in 50% fearful and 50% happy, we saw increased reaction time during the absolute ambiguity task for both younger and older subjects. And also the level of confidence. So participants were more unsure about the decision during the absolute ambiguity condition. And there were no significant age-related differences in both of the measures, reaction time and confidence level. We also found a positivity bias in the older subjects, such that during the absolute ambiguity condition, 50-50, participants tend to answer that the face was more happy rather than fearful. So in older subjects, participants felt that the face that they are seeing, the ambiguous face, is more happy. Regarding the first question of the neuroimaging study, we found that there was a higher locus coeruleus activity in the older subjects compared to the younger ones during absolute ambiguous facial expression. And this was also linked with slower reaction times in older subjects. So this shows that the older subjects' higher LC activity could potentially mean higher cognitive effort in deciphering the ambiguous facial expression. Next, to answer whether the locus coeruleus connects with the regions of the prefrontal cortex during the same task, we ran a generalized psychophysiological interaction and observed that the left locus coeruleus was significantly higher connected with the dorsolateral part of the PFC in the older compared to the younger subjects. We also did many controlled analysis as well, where we selected multiple regions of the PFC. So not only the DLPFC, but we also took the dorsomedial PFC, ventrolateral PFC, also anterior cingulate cortex, and also the insula, but only this was specific. The locus coeruleus connected with the DLPFC more in the older compared to the younger subjects. And most importantly, this locus coeruleus DLPFC connectivity in older subjects was linked with lower level of stress, anxiety, and also higher level of emotional resilience index that we did using the PR principal component analysis. So this shows that this LC-DLPFC coupling can potentially be very important for maintaining this emotional well-being in late life. To summarize, we found that we delineated the locus coeruleus, and we also found that the higher locus coeruleus activation in the older subjects. And this potentially can reflect cognitive effort in late life, as we see there was a link of higher locus coeruleus activity with slower response time in older subject. And finally, we observed that the LCDL-PFC could potentially be a very important adaptive mechanism pathway in late life that can further be investigated for intervention purposes using the TMS, like the magnetic stimulation. And with that, I would like to thank the lab members for their help in collecting the data, the participants as well for their valuable time, especially Mariam Ziai, the principal investigator of the lab, and additionally the co-supervisor, Professor Heidi Jacobs, her help with the Locus Coeruleus. And with that, thank you very much for listening as well. Okay. And you can feel free to scan the code for more information regarding what we do in the lab and also to read the preprint. Thank you very much.